Oh, no multiple choice, just free response for a few weeks. Yeah, always had to get used to it. Yeah. Is this kind of slow? Oh, it's cool. Starting a sec. Thanks. We'll get started in a little bit. I don't know what's wrong with this. Then all right. Let's see. It's still having problem. What is? Let's see. Morning. Zoom link. Zoom. I just need the review test. Okay, review test number 1. No. All right, let's take a look at this. You guys looked over the review test. Hi guys. Okay, we'll get started. I can't find the review test. Ay. This is what the heck. All right. Let's take a look at this. It's because I only have one monitor, so it's a little bit harder to go back and forth. Yeah. So I was hoping there's a better way to do this, because if I have 2 monitors, I can go back and forth very quickly. Okay. Is it kind of blurry? Yeah, it is, isn't it? Okay. This one, right? How do you guys feel about this test? Yeah, it's not too bad. Or it's a half of test. And this one has 8 questions, but we only need to, the new test has 6. Yeah, so 2 less. Okay, well, let's take a minute on this one. Let's go over it. So the first 2 are heavily weighted, they're 10 points each, yeah, on the new test. And there's only 8. So once again, question 1 and 2 are 10 points each, the lab related experiment. So it's very similar to what we have here. And then this is the test, right? I just want to make sure. Trichomes, yes. And then test question number 3, 4, 5, 6 are 4 points each. We're gonna keep with that scoring system. And so let's take a look at this one, okay? I'll have to switch back and forth between the 2 screens because I only have one. So for these kind of questions, especially 1 and 2, one of the things that we have to do is we have to understand the context, because it is not going to be something directly that you have seen before. So the AP tests have designed that they'll find a question that may be not as familiar, but biological concepts are similar. Yeah. So this one, for example, is about plants. Okay, and there's no plant in AP Bio curriculum, but they use plant experiment quite often because that's what we do. A lot of the experiments are done with plant. So they tell you what trichome is, even though you haven't seen it. They're little fuzzy hairs at the outside of the plant leaves, plant stem. So what they will do is that they provide protections. So essentially that they provide protection against being eaten. So you can think about this, they use the word herbivore, right? So you can think about trichomes are adaptive advantage for plants. Plants with trichome less likely to be eaten. So it's good. If you're a plant, you want to have as much trichome as you want. So it tells you also that trichome density is genetically determined. So meaning that high density trichome plants will produce high density trichome plant. So they're not influenced by environment, but they're genetically determined. Okay, so you can assume that. So here is an experiment. You want to talk about the stem trichome density within plant species. So what did the student do? They counted the number of trichomes in 6 plants in each of the 3 different populations. So you'll see the a little bit here. We have 6 populations, 6 different plants, 1, 2, 3, 4, 5, 6 in 3 different populations. Okay, so what that means is that if you look at population 1, the first plant, the density is 8. So that's how many trichomes per square centimeter per area, right? The second plant in population 1, it could be your backyard, for example. Population 1 is 11, 9, 10. So he took out 6 plants, that's it. In your backyard. The second one could be in the park. The third one could be in your front yard, whatever. So those are the 3 different populations. And then he measured the average trichome density. So it seems like on the average, population 3 has the densest trichome average, right? And if you look at the standard error of the mean, those are the error bars. So don't forget the standard error of the mean are those if you have let's say if you have let's say this is 6. So if the standard error of the mean is 1, whoops, what I mean is 6 here. If the standard error of the mean is 1, you're gonna get those trichome. The average density is 6, but the standard error of the mean is 1. You're gonna get those error bar that goes from 5 to 7. Okay, so that's what that means. So what that means also is that you can assume about 63% roughly of all the trichome densities falls between 5 and 7. So when you have plus minus 1 standard error of the mean, your data sets about 67%, 63% falls within 1. If you do plus minus 2 standard error of the mean, then more of your population, about 95% of the population falls within plus minus 2. So that will be from 4 to 8. So you can see that there is a bigger range. So more population should fall in 2 standard error of the mean. Yeah. So that's what standard error mean means. Okay. So hello, let's come in. We just started. We're doing the first question. Yeah. So the student used the data to calculate the mean trichome density, and the results are provided. Okay, so that's what this result. Does that make sense? The 3 populations are 3 different areas, and he only measures 6 plants. So if I were to critique this right up front, 6 plants is not a lot, right? You probably need to sample more plants to get a better sense. Okay, so we don't know what kind of sampling technique the student uses. All right, so the next thing is on the axis provided, create a label graph to illustrate. So you have to graph this. So one of the questions for AP response is that you have to graph. And it says they want 95% confidence, so plus minus 2. Right? So this is 1, right? So you want plus minus 2. Okay, so standard error of the mean is 1. So if you want plus minus 2, then you have to do plus minus 2, right? Okay, if the standard error mean is equal to 0.6, then what is plus minus 2 standard error mean? This will equal to 1.2, right? Double, right? So it depends on the standard error of the mean. So once again, if the standard error of the mean is equal to 0.4, so if you want plus minus 2 standard error of the mean, then that will equivalent to plus minus 0.8. Okay, so right, so that's what standard error of mean does. All right, so to do this, usually get few points here. 3 points usually for graphing of 4. So you can see from this graph, you just plot it out. Okay, so your y-axis would be the, I should really plot this out. So let's take a look at this. So let's just do this exercise together to plot this out if you haven't done so. Here, I'm gonna take a picture of this. And then we can plot this out on this one, right? Okay. Okay. So that, and we have to have our data, right? So again, it's a little bit harder to do digitally, but this year's AP is going to be in person, right? So most of the stuff is going to be in person. So make sure you can do this. I usually like to use pencils for AP tests, and for all my tests I use pencils. And then, but you can use pen. Just make sure you leave enough room if you were to, let's say, for example, come back to a question. If you don't leave room, it's really hard on the package to come back to any question. So leave enough room so you can always come back, okay? So that is the graph there, and then we're asked to, what are we asked to do? We're asked to graph this. So you can take the data. Here, and then you just plot it, right? So I'm gonna, might as well copy the whole thing over, okay, to this. Wish there's a faster way to do this, but because the lagging. So then I just put it here, and you can see the data and plot at the same time, right? Okay, so it's always better if you can see it. So if you have a small monitor, it's pretty small, okay? So usually plot, you don't have to give it a title, but you have to label the axis. So for example, if I simply put trichome density, then that is not a point, yeah? Okay, trichome density. I don't think that you're not gonna get a point because you forgot to put the units, so you have to put unit. So here you can see is the number of trichomes per centimeter square. So number of trichomes per centimeter square. So just keep in mind, you gotta have units. And the x-axis would be the populations, right? So this is your sample population or simply populations, yeah? And this would make sense to do bar graph. You're not gonna do a scatter graph. Usually the scatter line graph is versus time, so you can see how it progresses over time. Well, although you can do that with bar graph too, but this would be a bar graph too. That's the most common sense graph to use. So they want you to plot it, so I'm gonna plot the mean, right? So make sure you use as much space as you can. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's not good because I have up to what, 16, 14 to 16, right? So that would not be a good one by 1. So this would be 2, 4, 6, 8, 10, 12, 14, 16, and 18, and so forth. And 3 populations. So if you want the first population to be, let's say here, the average is 9, so I go to 9. Okay, so this is my population number 1. It's going to be a bar graph. And then you want 2 standard error of the mean. And the standard error mean we said earlier is 1. So 2 standard error mean will be 2. So you got to go up to 11, and you got to go down to 7. So you got to include that error bar. Yeah. If this is a bar graph, then you're gonna do like a bar graph, right? Okay, you can shade this if you want, but it will be something like this. Yeah. And if you look at the second one, second population, I'm gonna go to 11. So again, use all the spaces. Don't cram them together at one corner. So I'm gonna go to 11. So here's 11, and 2 standard error mean will take me to 13, and also go down to 9. So this will be the population number 2. Okay. And the next one is number 14. So 14, let me go to 14 is here, and go up to 16, go down to 12. And then you just draw the arrow, draw the rest of the bar. That's it. Okay. And every time you have standard error mean questions, it's really easy. You always want to look at to see if the error bars overlap. Because if the error bars overlap, like population 1 and 2, there's no statistical difference between them. So essentially, they are the same data. Yeah. So if the error bars overlap, then within 95% confidence that these two, population 1 and 2, have no difference in terms of the trichome density because they overlap. Okay. So the error bars are not supposed to overlap. If they do, then there's no difference. So you can say there's no statistical difference in our result. So when it says there's 9 on average versus 11, because the error bars overlap, they're very similar. So there's no really much difference, significant differences. Yeah. But if you look at population 1 and 3, there is a difference. 1 and 3 does not overlap. 1 and 2 does. 2 and 3 does. But 1 and 3, they don't overlap. The error bars, the standard error, the mean do not overlap. So there is definitely a difference between 1 and 3, even within 95% confidence, right? So that's what the error bars will tell you. So we plotted it as much to scale and labeled it. So that's easy, at least 3 points. Yeah. Okay, they won't tell you the point, but that's kind of how you guess. All right, next one. Based on the sample mean and the standard error of the mean, identify 2 populations that are most likely to have statistically significant differences in the trichome density. So the answer is 1 and 3. Okay, and your justification is simply that population 1 and 3 are statistically significant different. Okay, sorry, I bit myself and trying to recover. It was stupid. Have you guys bit yourself before and then you're like, damn, how dumb can I bite myself? And then like 30 seconds later, you bite it again. Yeah. Okay. But try not to do that. So it's so stupid. And it takes takes so many days to recover. Biting yourself. Okay. So anyways, so this is 1 and 3. Yeah. And your justification, once again, focus on the error bars. They don't overlap. That's why they're statistically significantly different. Okay. All right. Okay. So they use those stupid wordings. That's one thing I don't like about biologists in general. They use wording that it makes something simple very difficult. But that's what PAPI is. Okay. All right. I'm not gonna write it down. So I'm just gonna move on. Next one. Now, next one, describe independent and dependent variables. So when you look at an experiment, we haven't reviewed this for a while, so give me a little bit of time. So every time you have an experiment, you want to talk about, for example, hypothesis. Yeah. So they can ask you to state the hypothesis or no hypothesis, right? No hypothesis. So we'll come back to that as the first thing. The second thing, you may be asked to look at the dependent and independent. So dependent is always the y-axis. So in this case, the dependent variable will be the trichome density. So I'm going to label it as dV, dependent variable. And the independent variables will be the x-axis. So they're independent. So that would be the population number, the location of the population where you get them, right? And you may also be asked about the control. So once again, the control eliminates Well, the control allows you to see the effect of the experiment. And between your control and your experimental group, there can only be one variable that differ. So you cannot have multiple variables. The control allows you to see the experiment. So it is important because without the control, we don't know what our data means. Yeah. So for example, if you have this versus that, right? So without a control, what will be these two experiments? We only know that we have no reference, right? So what if the control is like this? Then both experiment 1 and 2 is high relative to the control, right? Because we know the control. But if the control is absent, then we don't know what that means, experiment 1 and 2, yeah? And what if your control is up here? Then it turns out now, in this case, experiment 1 and 2 is lower. So without a control, we cannot talk about the experiment. We don't know what our experiment means. There's no reference. You don't have something to compare with. So that's the importance of a control. So I want you to keep in mind, they'll always ask you about the control. Okay, then so those are probably the most important thing. Okay, when you look at the experiment, of course you have to know how to gather data. Okay, sometimes they don't ask you for that, but right here we'll look at part C. So we also do a bunch of things. So all these are points. You want to describe, talk a little bit about it, not to state. State, you just write down the answer. Describe, talk a little bit more. Dependent, independent, and control. And what do you want to test? So here's your hypothesis that you're testing. That higher trichome density in plants is selected for in the presence of herbivores. Yeah. So when there's herbivores around, if you have higher trichome density, then you survive more than lower trichome density plants, right? Okay, especially in the presence of herbivore. Okay, and you also have to do, so that's at least what, 3 points, right? Because you have to do 3 things. Identify appropriate duration of the experiment. So how long does the experiment have to last? Is it seconds? Makes sense. That wouldn't make sense, right? Is it days, weeks, right? To ensure that natural selection is measured. So natural selection takes at least several generations. So you want to think about, okay, for this plant, what are the several generations, right? And predict the outcome. That's support. So sometimes they ask you to support the hypothesis that indeed having more trichome density is selected for, and without trichome density, you select it again. Sometimes they ask you to refute, right? Usually to support, yeah. Okay, so those are the things that you have to address. So that's the majority of the point. Okay, so what do we have to do? Okay, so what is the dependent, independent variable here? Okay. So let me show you the scoring guide. And with the scoring guide, you can kind of give a sense of what exactly they're asking for. The scoring guide sometimes is very confusing and there's not enough detail, but they give you some guidelines what you can talk about. Okay. So again, free responses questions, they're released online. So let's see what they did here. So the graphing is 3 points, right? You create a graph. So look at the 3 points. You have correctly labeled, scaled with units, and then you got to do a bar graph. And then you have to make sure you plot the standard error of the mean correctly. So 2, okay, above and below the mean. So that's 3 points. Usually graph is just 3 points. And part B, we're going to talk about this. So we do part B already. We did, right? Yeah. So population 1 and 3, and your justification. Now, if you look at the points assigned for justification, for example, they're just saying that the error bar for 1 and 3 do not overlap. That would be fine. But why is there another bullet? So that's something else you can talk about too. Yeah. So you can say that this is a fancy way to say it. So the mean plus minus 2. So that basically is population 1, right? And is less than the mean minus 2. So the highest for population 1 is still smaller than the lowest for population 3. Yeah. So the highest for population 1, that's why it's +2, is still less than the lowest for population 3. So I don't like that. Just say that they don't overlap. Yeah, this is making something simple more complicated. But again, that's what that means when you have multiple bullets. Yeah. Okay. So let's look at this. The next one. The next one is that dependent, independent. Okay. So if you look at the scoring guide here, they're giving you 5 points, and that's kind of what we were guessing as well, because independent 1 point, dependent 1 point, control 1 point, identify duration 1 point, and predict the result that supports the hypothesis. So 5 points. So the way to put it like this is a little bit confusing because it depends whether you are going for this one or this one. Yeah. So for example, if I were to do this experiment, okay, I would look for, okay, what is your control? For example, my control, this makes most sense to me. My control will be absence of herbivore, right? So this would be my control. So I have to say control has no herbivore. And then my experiment will be here. That's it. So I have 2 groups, yeah. And this experiment will be a population somewhere. We don't have to specify, but and then my dependent variable, which is my y-axis, okay, would be trichome density. So this would be the trichome density, yeah. So what that means is that okay, and I would do before and after. So before means time 0, and after means after a certain amount of duration. So I'm gonna say my duration is like weeks. At least there's several generations. Yeah. So you see the duration. They even take points for just saying more than 1 generation. That's stupid. Yeah. But you can just say weeks, as long as there's multiple generations allowed. But you would actually have to do before and after. So let's say it's like this. Your control, okay? So before they all have the same trichome density, right? Your this is the with herbivore, okay? Your experiment has herbivore, this one has no. Okay, so that would be a time 0, okay? And after, let's say 3 weeks, and how is my graph? How is my result? Yeah. How would the so this is number of plants. This is trichome density. Let me see. The trichome density here, present herbivores. Okay, so yeah. Okay, so then you count the plants again. You will see something like yep, the trichome density would be higher relative to control. So I don't. Let me see before and after. Let me see what I want to do before and after. Okay. So afterwards, if I go count it, this experimental population, the average trichome density will go up. Yeah. And control, there's no herbivore, so there's no difference. Yeah. So maybe this would be about the same. Okay. So it'll be something like this: that after a certain amount of time with herbivores, so all the low trichome density plants are eaten, so your average is higher, right? So you're gonna have a higher average, okay, compared to the control, okay, after the time. So even if you don't plot the initial, it's fine. I was thinking about I should plot the initial trichome density, but I guess you don't need to. Okay, so this will make sense because if there's no herbivore, there's no reason for the trichome density to change since the trichome density are genetically determined. Okay, you shouldn't change that. But with the herbivores, so the lower trichome density are eaten after 3 weeks, right? So the survivors have higher trichome density, genetically passing them on to their offspring. So the survivors will have higher trichome density than before, yeah? So we're calculating the trichome density. So I'm actually following this column, yeah? So my independent would be the x-axis, so would be the populations. This would be the presence of herbivores, okay? And my dependent, the y-axis, would be the density, the average trichome density for the populations. and my control is no herbivore. My experiment is herbivore, right? And then duration would be several generations. I said about several weeks, enough for many generations to pass for natural selection to act on. And then the prediction is that there's the experimental group should have an increased average trichome density because the lower trichome densities are eaten. So that would be 5 points there. Okay. So it's a little bit hard to transfer this over, so I'm just discussing it. Yes. Yeah. Yeah. So the question is that for these kind of questions, do we need to restate the hypothesis, right? In this case, for this one, you don't need to, but a lot of times you do need to. They do ask you about what would be the hypothesis, or what would be a result that's like this one is what is the result that supports the hypothesis? Because we already know the hypothesis. So if you don't have enough time, I wouldn't restate it. But when I talk about this, this is my prediction, right? So when I talk about this, I would have said that in my experimental group with herbivores, the average trichome density is higher after several weeks. That would be my prediction. And I don't have to say, therefore, this supports the hypothesis that higher trichome density plants are selected for. That would be kind of redundant, yeah. So I'm basically just answering the question, yeah. Okay, but sometimes it is very important. You have to keep in mind what is your hypothesis. What if I want to, what I don't want to, what about against the hypothesis? What if you want to predict the result, and then your hypothesis would be wrong, right? Then your control will be like this, and afterwards is lower. So this is your herbivore, right? So this is saying that after 3 weeks, your average trichome density, you have less trichomes, right? Less number of trichomes. That means all the high density trichomes are eaten by herbivores. Then something is wrong, right? So the initial, your hypothesis that higher trichome density protects the plant, that is completely wrong, okay, based on your result at least, right? So you would have to argue against or support, so always keep that in mind, okay? So I'm not going to go over the second one, so I basically, this is the first thing came to mind, yeah? But I guess this is another way to look at it, okay? So let's move on to the next one. So is that it? I think that's it. So there's 10 points, yeah, for the first question, okay? All right, so let's look at the second one. Yeah, the second one, we might as well do this. Now, if you get an evolution question like this, cladogram, then you're blessed for that year, because evolution questions, phylogeny, cladograms are amongst the easiest question on AP Bio. That's what you want, yeah? So you're hopeful to get one. So let's look at this one. This a little bit about immune system. It says mammals have milk, right? That's one of the trademark characteristics of mammals. We produce milk, and that is adaptive because mother, as soon as the babies are born, mother does not have to go out look for food and leaving the young by themselves, for predator. So they can protect their young while having food. Now they also have antibody. This is another beautiful thing about milk. So the antibodies are proteins produced by mom, which will be transferred to the baby through milk, so via milk. Okay, so that's a beautiful thing. So this antibody will now protect the baby, okay, against viruses and germs and bacteria that baby has not been exposed to. Okay, so this is why breastfeed is the best, breast milk, right? Because if you use formula milk, there's no antibody protection. Yeah. Okay. So you pass to the offspring. Great. So mammal milk also contains sugar. So of course that's lactose, not very sweet, and also proteins, of course, A, B, and casein. So they talk about this. So if you didn't know about it, again, they'll give you additional information. In this case, they tell you what is inside the breast milk. So we got 3 different type of proteins and sugar. And if you look at a cladogram, they're trying to talk about cat, cow, horse, humans, pig. And they talk about these characteristics. That's why this is the cladogram, yeah? Trying to show you the evolutionary history. So you can see that from the look of this, looks like humans and cats They are more related in the sense that they neither of them have protein B or casein. And cow, horses, and pigs, they have all of them. So based on that, what do they want? Yeah. So if you look at part 1, using the data, construct the cladogram on the template provided to indicate the most likely evolutionary relationship between different mammals. So cladogram. Indicate on the cladogram where each characteristics arose. Where did it pop up? So that's 1 point. Cladogram is 1 point. And then justify the placement. So talk about it. Why do you put it there? Okay. So that's it. So this is super easy. This is like one of the easiest questions you can get. Now let's just look at one side at a time. Let's focus on the left side. How about that? Okay. So you could say that again, we established that cats and humans are more closely related, and cow, pig, and horse. So either one is going to be the same. Yeah. Okay. So let's look at the left side. So everybody has lactose and protein A, so this shows up in the common ancestor before the common ancestor to all 5 animals. But protein B and casein are found in cow, pig, and horse only. They're not in cat and mammal, so that'll be fine, right? So wouldn't that the most common sense one, right? So again, if you forgot what the data is, you can just, I'm going to zoom out a little bit. If you look at the data, once again, cats, humans do not have protein B and casein, cats and humans. So protein B and casein arose before the common ancestor, giving rise to cow, pigs, and horse. But there isn't such derived characteristics for cat and human. Okay, and so that's it. It's super easy, right? Now, you can also do it this way. So you can lose the characteristics of casein and protein B, and then therefore cats and humans do not have casein and protein B. So you will put all the characteristics here in before the ancestor for everyone. But this would be this, yeah, loss of character. So you can lose characteristics as well, yeah. Like a lot of times, if you have reptiles, right? Reptiles, and if you branch out to snakes, then you will lose a characteristic. What characteristic is lost for reptiles to go to snake? Would be legs, limbs. Yeah, okay, so they don't have limbs. So that would be a loss of characteristic, and that is fine too. Okay. Now, if I were to ask you to identify a clade, so then a clade would be the common ancestor and all the descendants from that same common ancestor. So the clade that include cow will be this one. So it will be the cow, pig, horse, and the common ancestor that includes cow, pig, and horse. And this is another clade that includes cats and human. You cannot just, this is not a clade, yeah? So horse and cat, if you want horse and cat belong to the same clade, then you gotta go far back. This will be the clade that include horse and cat because it has the common ancestor and all the descendants from that particular common ancestor. So since if you want to include horse and cat, then your clade will be the whole thing. So a clade that is the common ancestor and every single descendant. You cannot miss one from that common ancestor. So you got to include all that to be included in a clade. So that could be another type of question that you asked to do, all right? So super easy, right? So now what else do we have to do? We have to justify. So I thought we already did, but specifically you justify. So if I use this one because it makes more sense for me, okay? I'm justifying this because the casein B, protein B and casein arose only in the common ancestor that gave rise to pig, cow, horse, and not to cat and human. So that's your justification, yeah? Okay, all right. So 3 easy points. Evolution questions are super easy, so please don't freak out. They'll give you a lot of data. It feels like super hard, but they're super, super easy, yeah? Okay, now part B has to do with immune system. Now we did immune system before, but here, part B says this, and they do this quite a bit. Describe, okay? 4 steps in the activation of mother's specific immune response following exposure to bacterial pathogen, and predict how mother's immune response will differ upon second exposure to the same pathogen 1 year later. So there's several things here. Let's just take one step at a time. So first one is you got to describe the activation of mother's specific immune response. So tell me what I did wrong. If I start talking about fevering, if I start talking about white blood cells destroying the bacteria, if I start talking about inflammation, I get no points. Why is that? White blood cell eating up the bacteria isn't an immune system. We will get no points whatsoever because the question specifically asks for specific immune response. So specific immune response only involve the B cells and the T cells. They are not your skin. They're not your sweat and enzyme, digestive saliva. They're not your white blood cell that circulates like macrophage and neutrophils. They're not inflammatory response that brings more blood supplies to the site of injury so that they can be fought off, the bacteria can be fought off. So none of those. So you can't talk about any of those. So it has to be specific immunity. The next thing is that when you talk about the specific immunity, they say 4 steps. It might as well be 3 steps, 5 steps, 10 steps. It depends how many steps you want to put it out. So there's a bunch of steps, yeah? So look at all the bullet points. Okay, you can talk about this in 5 steps, 6 steps, 4 steps. So they just wanted to put 4 things, okay? And in a stepwise fashion, okay? So don't worry about how many steps. So you can think about activation of specific immunity in 3 steps, 4 steps, 5 steps. There's no relevance here, okay? So this is the thing that the AP wants you to do. So if you don't remember specifically what happened, it's okay. But you should at least know some basic stuff. So let's do that quick review. And we talked about immune system several times in the past. But if you look at the basic review here, let's see what we talked about here. Yeah. Okay. Let me get rid of the screen color. And this is just immune system. Okay. I'm going to do a very quick branch of the immune system. Let me just get rid of this. Okay. So again, your immune system, there's innate. Innate is non-specific. Innate immunity. And there's also the acquired. Okay. So this is the acquired or adaptive. So there's different way to say it. Yeah. These 2 ways. These are specific immunity. Okay. So that means they target specific pathogen. So the acquired immunity is what ultimately will protect you. And then there's 2 branches. There's the humoral immunity that involves the B cells. So selection of a B cell into proliferation. So you get a bunch of plasma cells, and these will release the antibodies. Yeah, and some of these antibodies will be passed from mom to baby. There's also the cell-mediated immunity. The cell-mediated immunity involves the T cells proliferation to become the cytotoxic killer cell. So what they want you to differentiate between these 2 specific immunity is that the antibodies, they circulate in your blood, they target the antigen, which means the virus and bacteria, COVID, for example. They target antigen directly. They will bind to the antigen, neutralize them, agglutination, and also phagocytosis will follow. So they target the antigen, the viruses directly. Okay, so they mount attack directly, but not the cytotoxic. These ones do not target the pathogen. These ones, they kill our own infected cells. So because infected cells are factories for virus reproduction, right? So they go after the ability to reproduce. So that's the specific branch. And in order for the 2 branches of the specific immunity to work, we need the T helper cell. Yeah, we need to activate the T helper cell. And who activate the T helper cell? We have a bunch of dendritic cells. they present antigen. So they're antigen presentation cells. Antigen presenting cells. Those are one of the most important cells. They're everywhere in your body where potential antigens can enter. So these are called APC cells. So the purpose for dendritic cell is that they are exposing themselves to antigen. They will eat up the antigen, spit it back, the antigen fragments on their surfaces, and then they will present them to T helper cells, thereby activating the T helper cells. Once the T helper cell is activated, T helper cells will activate the humoral immunity as well as the cell-mediated immunity by releasing chemicals, okay? And these chemicals are called interferons, yeah? Okay, or interleukin, not interferons, I'm sorry, interleukins. So these chemicals are chemicals that immune cells talk to each other and activate each other with. Remember the nervous system we used was dopamine, not dopamine, it was neurotransmitters, like dopamine, like serotonin. But in the immune system, these are called interleukins, yeah, okay? So we already talked about more than 3 steps, right? You can say that dendritic cell presents, these are antigen fragments on their surfaces. That could be step 1. Dendritic cell activate T helper cell. That could be step 2. You can also say T helper cell activate the B cell. That could be another step. Okay. And once the B cell is activated, they always get a memory cell, memory B, for second exposure. That's another step. And once the T cells are activated, they also produce a bunch of memory T cells in case you get a second exposure. And that's the basis of a vaccination, right? We want those circulating memory B cells and memory T cells. We want them to be in our body, circulating so that they can quickly become reactivated upon second exposure. That could be another step, right? So these memory cells will very quickly become plasma cells upon second exposure, right? So you can also talk about the plasma cell generate monoclonal, one clone antibodies that will target specific antigens and therefore neutralize them. That could be another step. So any one of those steps will be the 4 steps and a little bit description. So look what they give you points for. You can say endocytosis of the virus, which is antigen, by dendritic cell, okay? Macrophage is also antigen presenting. B cells are also, there's 3 antigen presentation cells. So you just have to know dendritic cell, okay? You can also even say the degradation of antigen by the macrophage. It's so stupid, I wouldn't even put it, yeah? An antigen complex with AMC, not AMC, AMC is a stock symbol. So this is a MHC, major histocompatibility complex. This is your self marker, yeah? So this identify your own cell. So that is detail, okay? So you can say antigen bind to self markers or MHC receptors, presenting. So all this is about antigen presentation, yeah? Okay, and then what? Then you can also talk about, there's so many things you can talk about. So the only way you don't get points is by talking about nothing, yeah? We can talk about anything here. So you can also talk about that the T cell, right? T helper cell, recognize the antigen, okay? And you can say activation of the T cell. You can also say that the T cell release chemical. Those are the interferons, interleukins that activates B cell. There's a bunch of things. You can also say activate the B cell will clone itself to get a monoclonal antibodies or cytotoxic killer cell. You produce antibodies. You can even say that. Antibody recognize antigen. You get points by saying that. Antibody bind to the antigen. Yes. You can also talk about memory cells are produced. So anything you want. You just know something about immune system, you get points for, yeah? So you can never miss points on immune system because there's so much you can talk about. So that's that. And next thing is prediction. We have to do a prediction. So how mother's immune system will differ upon second exposure. So they want to know the difference between first and second exposure. I think I gave you 4 differences. So let's go back and review that. What are the 4 differences between primary, that would be the initial exposure, and the secondary exposure in terms of in terms of this graph. This is the time. This is the number of antibodies that matters. Okay. So your first exposure is always a much smaller bump, and it takes longer, right? And the second exposure, so this will be the first exposure, and the second exposure is always much, much faster. And the amount of antibodies is, this is a log scale. So the amount of antibody is hundreds times stronger, yeah? So it's gonna shoot up. And then it's also long lasting, kind of like that. I'm being a little bit more exaggerated here. Okay, so it's not just 1 or 2 times greater antibody load. The second exposure is 100 times more antibody. Okay, and right now we're debating whether the antibody will disappear. That's why we get booster. So if it does drop, then we get a booster shot to get antibody amount boosted. Okay, so what is the difference? So the second exposure is, these are the characteristics, is faster than first exposure, of course. Okay, and also much greater antibody load. So that would be another characteristic. So much greater antibody load or exposure release, right? And then it is also, okay, involves memory cells. So the memory B and memory T. Okay, and lastly is long lasting. You can see the primary exposure, the antibody counts drops back to nothing. Okay, so it's longer, long lasting in circulation. Yeah, so those are the 4 things you may talk about every time you have questions comparing first and second exposure. Okay, so this is why second exposure, we don't even feel it. Okay, even if you got exposed to virus and your antibody is going to fight it off without any symptoms, so you don't even know if you had it. Okay, so that's it. So you can talk about those 4 things. So predict, how is it different, right? So you can see the prediction, much more rapid, memory cells, greater production of antibody, lasting longer. And you can even say the last one, which I did not talk about, antibodies has greater affinity for the virus, for the second exposure, because those antibodies are very specific. The first exposures, your antibody is a little bit different type of antibody, but that's just detail. Let's look at number C. So let us see what we're trying to do here. We're trying to predict what is the likely consequence of nursing infant who is exposed. Yeah. So talking about nursing infant who is exposed to intestinal bacteria to which mother was exposed 3 months earlier, and justify. So 2 points here. So if the nursing, so that means the infant is drinking mother's milk and the mother had been exposed prior, so the baby should be protected, right? So the baby should not be sick, right? So that's common sense, right? And then your justification, of course, you're told in the beginning that antibodies are passed to infants through breast milk, and therefore become offers protection for the baby. So that's like 2 very, very, very easy points. And if you decided to skip this one, then you will be at a loss, because you can even say that the baby will get sick and even die. Okay, so that is definitely a possibility. That just means that there's still not enough protection despite the passing of antibodies to the baby. So there's like any answer basically, as long as you can justify it using biological concept. It cannot be fancy. Like the concept that doesn't work has to be biological concept. So baby could die because the antibody does not work, right? Or not enough of it. So there's that. Super easy, right? Okay, so they're much easier than we think. Okay. Now question number 3 is a stupid question. I'm just going to skip altogether. It's about ecology. It's not a very good question. Yeah. We'll come back if we have time. Okay. I think we have time. So that's question number 3. Let's look at next one. What is question number? Give me a sec. I like to look at just the question, not the scoring guide. let's take a look at this one. So we're done with question number 1 and 2. So those are the most heavily points. In the rule of thumb is if you get stuck on, let's say, one part of a big question, like question number 1, you need to move on. Yeah. So there's a lot of mistake people make is that they got stuck on one point. They couldn't make the prediction correctly. And then they spend 5 minutes, 6 minutes on one point prediction and refuse to move on. And that will be a big mistake because you want to get to the later questions, which are much easier to get points on. So you never want to get stuck on one question and get wasted on time. If you look at the in terms of the time, in terms of timing, they took out, so the new AP has 6 full response instead of 8. So they already give you a lot more time now. So this is 90 minutes, right? So 90 minutes, they will have a 10-minute reading, which is very stupid. When you read, you can take notes, but you cannot answer the question. Okay, so 10-minute reading. So that would I would spend that 10-minute reading, look for which question is most difficult. Okay, so that you want to pace yourself correctly, right? So if you think about these are 10 points, and then the rest of them are 4 points. So this would be 20 plus what is 16. So 20 plus 16 is 36 points, right? And then the 90 minutes minus the 10 minutes reading would be 80. Then you can divide it by 36 points. So this would be how many minutes per point, right? So if you divide that, 80 divided by 36. So that would be 2.2. So that means the first question is about 22 minutes. And these are about 8.8 minutes, maybe 9 minutes, yeah? So you don't want to spend like 6 minutes, 7 minutes on one point. It's not worth it, right? You want to make sure you get the other ones addressed before you're coming back to this. So, and another problem people make is that they roll way too much for 1 point. It turns out to be an essay, a lot of repetition of the question. So people write about predict one ecological consequence, for example. They'll say one of the ecological consequence of a local plant community that is likely. So you just write the question again. Please don't do that. It's just a waste of your time. Yeah. So you want to save time, get to the point, answer the question. You only BS like that when you're not sure what to do. But even when you do that, don't repeat the question. Yeah. Never rewrite the question again. That's just it's waste your own time. Yeah. Okay. So the reason I don't like this question number 3, once again, I just want to talk about this is ecology. It's kind of common sense. Now, when you are building a sports complex, it is always bad for the environment. Okay. You could argue it's bad for the environment, but you can also argue this is good for the environment because now human activity will come in and do maintenance, right? There will be water, there will be fertilizers, there will keep hopefully animal diversity. But initially, when you do the urban development, you gotta clear an area, you gotta clear a forest, for example, you're gonna get rid of biodiversity for sure, right? So this ecology question always had to do with biodiversity, yeah. And it could be natural selection if they want to connect to it, biodiversity, and any kind of human activities like urbanization, deforestation, logging, you know, we do farming. All these are bad for the environment, yeah? Because even farming, you can overuse the fertilizer, which causes eutrophication of the nearby ponds and lakes. You can also have loss of topsoil if you over harvest, right? So you can also, you just have a whole bunch of problems. You can overuse insecticide, pesticide, those are chemical pollutions, right? It's just anything with human, it's very easy to argue humans have done bad. You can talk about pollutions, right? All these things. So what they're saying is predict one ecological consequence on the plant community and then justify it. So it's kind of stupid. You know, if you can just, we'll look at the answer why this is, you know, And then to maintain the playing field, large quantities of water, that's also a bad thing, right? Using water and chemicals are applied. Predict one effect on the local animal. So you gotta talk about the animals now. Okay, this could be bad for the animal or good for the animal. Yeah, we don't know. So either way is fine and justify. So it's like not much biological concept here. And then so if you look at the choices here, okay, what are the consequences to plant and animal, right? So if you look at question number 3, so here, so why is it bad for the plant? You can say the plants will die, plant death, loss of habitat. You can also reduce the number of species, okay, loss of biodiversity, lose genetic variation, habitat loss, they're all the same thing, yeah? And your justification, because to build a complex, don't you have to remove trees? That's why the plants die? Yeah, okay. And then you're changing the habitat, so change the amount of sunlight, okay? Your destruction by having building equipment there, you want to build a complex, right? You need a lot of building equipment and materials, and then you get rid of species. So it just kind of doesn't, there's no biological stuff here, just whatever you want to say here, yeah? Okay, you can, so what about on animal, right? So this, I don't think you'll ever see these kind of questions again. This is a little bit earlier, 2014. That's why these kind of questions are a little bit weird. They're more focused now, okay? So that's why I didn't want to worry too much. But all these terms you need to know, okay? Loss of habitat, same thing. You know, the animals will die or migrate to some other places. Loss of animal diversity. There can be mutation caused by chemicals. We don't know, right? Now, if you want to argue this is better for the animal, you can say that there's improved condition for animals. Then you just have to justify for it. So maybe there's more food resources. It depends what kind of animal, right? Other animals may come in because you have new habitat. Maybe you build a park that allows some animals now. Before they wouldn't be able to come in due to competition. Now they could because the man-made pond or park, right? All right, so it's kind of stupid. That's why I wanted to skip this question. Yeah, let's go to question number 4. Okay, let's see what that is. Question number 4. Sorry, I'm trying to scroll down. Take a little time. All right, so this is question number 4. Okay, so question number 4 has to do with this graph. Alrighty. So this is, let me shrink down a little bit. So let's look at this. This is a good question. Yes. Okay. So question number 4. And when you read this, guys, on the test, you get to mark stuff that you read. I would do that, but I wouldn't highlight everything. Then there's no point of marking them. And if you use a pen, please be careful. I have students, they mark so much, they couldn't read it anymore. That would be so dumb, right? So adult male guppies exhibited genetically determined spots. So the number of spots is determined by genes. Juvenile and females, they don't have spots. Yeah. So kind of Dalmatians. Yeah. When they grow up, the spots come out. And the female have no spot. So in the study of selection, so that's natural selection. So if you think about natural selection, you should think about everything that Darwin talked about, right? There's variations and there's adaptation, competition, differential reproduction, right? Survival of the fittest, right? And there's also directional selection. You favor more spots. And there's also disruptive stabilizing selection, okay? So in the study of selection, male and female guppies from genetically diverse populations, so that means a population with a bunch of different spots, were collected from different mountain streams placed together in an isolated environment that has no predators. So right now there's no predator put together, okay? The population was maintained for several generations in the isolated area before being separated into 2 groups. So again, we grab a bunch of guppies, male, female, juvenile, whatever. We put them together in an isolated environment that has no predator. So it's about predation, right? And it's genetically diverse, so we can assume there's a bunch of spots, okay? All right? And then we do this. One group was removed to an artificial pond with a fish predator. So now it's predation, yeah? And the second group removed to a pond with no predator, so the effect of predation. The 2 groups went through several generations in the new environment. At different times during the experiment, the mean number of spots per adult was monitored. So you count how many spots. So that would be your y-axis. the number of spots is your dependent variable, and your x-axis again is no predator and yes predator for the two groups. Okay, so simplify slide is one of those again. And then vertical bars represent two standard error of the mean. So two standard error of the mean. Okay, those are the vertical bars. So let's look at the graph. So if you look at the graph here, alrighty, so this is what we see. So first, if you look at this, okay, you always want to look at the y-axis, which is the average number of spots for adults. Only the adults have got, male adults have spots. And then the x-axis is time, yeah? So you can see right here, the population is split. So they're split here, and then predators present. So this is predator, okay, and the top one has none. So you can see that predators are selecting against guppies with spots. Yeah, that's why the average number of spots per male guppies drop, because all the spotted ones are eaten. The more spots, the more easier they appear to the predators. They're discovered more easily and more quickly, so they'll be eaten. So the number of average spot dropped. That's what the predator do. So we can know, we can look at the graph, and the first thing is the predator are selecting to eat the guppies with more spots. And that's why the average number of spots drop compared to the control. So this would be, no predator will be the control then, because the only variable is the presence of the predator. Okay, all right. So that's basically our conclusion by looking at this, yeah? So the question is, describe the change in genetic variation between 0 and 6 months. So the 6 months is right here, is right before the population was separated. So between 0 and 6 months, we're asked to talk about the genetic variation. Provide reasoning. So what is the genetic variation between 0 and 6 months? So how do we know? Yeah. Okay. I'm actually looking at the arrow bars. Look how big the arrow bars are. At 2 standard error mean, this means that we're 95% confident that the number of spots ranges from 8 to 12. Yeah. So that means all the fish that we brought into this controlled, isolated environment, there's fish with 8 spots, 9 spots, 10 spots. Even though the average is 10, there's a bunch of variation. So the width of the arrow bar represents variation. If the arrow bars are very, very, very narrow, right? If the arrow bars are very narrow, that means there's not much variation. Everyone is the same as the mean. If you look like this, then pretty much everybody have 10 spots. But when you have a huge arrow bar, that means there's 8 spot, 9 spot, 10 spot, there's 12 spot, so there's lots of variation. So the size of the error bar also indicates genetic variation, phenotypic variation. Yeah, that's the answer. So how do I describe the change? Yeah, remember, you want to describe the change. So the change is between 0 and 6. Look, I have a bunch of variation at 0, and at 6 months, look at my variation. Something happened, right? Then variation dropped. So the change is the change. Yeah? So the genetic variation in the number of spots decreased between 0 and 6 months. That's it. Okay, dropped a lot. And what would be the reasoning for that, right? So remember, this is enough time for multiple generations to occur. There's no predator yet, right? So what is causing genetic variation to increase? Well, the genetic variation drops, the number of spots on average increase. Okay, so be careful of the difference, right? We're looking for the change in genetic variation that dropped, but the number of spots on average increase. Well, this is sexual selection. That's the only reason. That female are choosing males with more spots. That is why after several generations, the average number of male spots increase, right? Because they get to survive and reproduce and pass genetically determined number of spots to their offsprings. So the more spotted male reproduced more and passed more spotted trait to their offspring. So the first 6 months, you can see sexual selection at play, non-random mating. is another way to say sexual selection. Yeah, that is why the number of spots increased, but so the total genetic variation dropped as the error bars shrunk. Yeah. Okay. So that would be the answer for this one. Is that okay? Right. Okay, we got this, right? So you see that do you have to know a lot to answer this? So far, our AP question does not require you to regurgitate any kind of stuff, right? Not a lot. Okay, and then part B. So propose one type of mating behavior that could have resulted in a change in the number of spots per adult guppy between 6 and 12 months in the absence of predator. So here's, we gotta be careful a little bit because sometimes you have to read the question several times. What the heck did we just read? Yeah. Sometimes when I read it, I have no idea what I just read. I have to slow down and kind of slow down a little bit. So we're looking for mating behavior, and we're looking for the spots, the change in the number of spots between 6 and 12. So that's that would be this. And I'm looking for the absence. So that would be the top one. I'm looking for that. Okay. The bottom has predator. I'm looking for absence and make sure I'm reading it right. So it looks like sexual selection again. Okay, again, A is not talking about sexual selection. Yeah, A is talking about the change in genetic variation. It decreased, and the reasoning is because the standard error mean shrunk. Okay, and B is talking about sexual selection. The mating behavior is sexual selection. So what exactly is the behavior? You have to state specifically that the females are choosing more spotted males for mating. So there's a selection, sexual selection for more spotted males. Okay, that's why the average number of spots increased from 12 to, I guess, 13. Yeah. Okay, so let's look at the scoring guide for what they say. Yeah. So this is question number 4 again. So part A. What is the answer? Yeah. Basically, genetic variation is decreasing, and then standard error, the mean got smaller. Okay. So that's all they want. 2 points. It's like so easy to get those 2 points. Yeah. Okay. So remember the error bar also talk about genetic variation. Yeah. And part B. What is one type of mating behavior? Well, then you see that sexual selection for males with more spots. That's it. Okay, now you can also argue, people who want to BS, you can say, oh, well, just by chance. So random mating behavior result in increase in number of spots by chance. Even that's that you don't have to identify the most likely reason, as long as any reason will work. Yeah, just propose it. You don't have to justify it, so don't try to explain why there's increase. Yeah, so just propose it. And then it's just the fact, you just state the fact. You don't have to say why the female loves male with more spots. So if you are trying to go into the reason why female loves males with more spots, then you're wasting time. Okay, they are not asking you to explain a reason why more spotted males are chosen. Okay, they're not asking you to justify that at all. Okay, we don't know how to justify that. Okay, so that will be it. Again, answer the question and move on. Okay, and plus C, propose an evolutionary mechanism that explains why the change in average number of spots between 6 and 12 in the presence of predators. So when they say evolutionary mechanism, so what are the evolutionary mechanisms, guys, in general? Okay, when you see that evolution mechanism means what causes evolution, right? What causes microevolution? We talked about 5 reasons. So mutation could cause this. Can mutation cause this, right? Or, okay, whoops. Natural selection can cause this also, right? You can also have gene flow. The population is isolated, so it can't have gene flow. Genetic drift is by chance, maybe. There's also non-random mating, right? So all these are evolution mechanisms. So they want to know what happened to the population with predator. Okay, so this is about natural selection. The predator population right here, you can see that the average number of spots dropped. right? That means the predators are eating, hunting, selecting males with more spots. So it's a natural selection due to predation. Predation is the selective pressure. Because of predation, okay, the predators, so you want to throw the word natural selection in there, proposed, right? So I would say natural selection, okay, is the mechanism, right? As a result, due to predation, right? Predation. Predators are eating. If you don't have to use fancy word, you can say that predators are eating males with more spot. That's why the average number of spot dropped. So there's a selective pressure. Okay, or you can even say that the male with more spot are selected against in the presence of predator. That's what I would put. Yeah. Okay. That's it. Males with more spots are selected against in the presence of predator by natural selection, if you really want to put that in. Okay, so you want to use those evolutionary vocabulary as much as you can. That's it. So let's see what the points will give you for. Let's see, this one will be all right. So they say we go a little bit. So natural selection is used in the context you get points for. You can say directional selection against individual with large spot. That's what we said. Right, the male with a large number of spots are selected against by predator. Right. You can even say genetic drift. Again, those are the mechanisms that resulted in decreased number of spots by chance. Okay, chance event. But you cannot say migration. There's no migration here. You cannot say mutation. There's no evidence for mutation. Those would be just fancy reasoning. So you have to say at least something that is plausible, and there's no right answers. And that's the kind of AP question. There's sometimes nobody knows what the right answer is. You just have to do the best. Yeah. Okay, number 5. Number 5 is also one of those questions you don't need anything to do. It's talking about GM crops. So if you go to the supermarket, you can see some of them will label GM. It's by law. If they're genetically modified, they have to label it. Yeah. So you can decide if you want to eat it. Okay. So they produce protein. So great. You modify them to produce protein that makes the plant resistant. That's great, right? So the crops that are genetically modified will be naturally resistant. I don't have to spray pesticide on them anymore because they can make the protein that kill the bugs, whatever. Yeah. So other genetic modification make plant more resistant to chemical. You can also do that. So you can have the plants modify their DNA to make proteins so they're resistant, or they become more resistant to chemicals that kills everything. All right. So A, common sense. Describe 2 potential risks of large-scale cultivation of genetically modified plants. So what if you decided to human, right? Large-scale cultivation. So you're going to grow the genetic modified plants in huge quantities. What would be 1 or 2 biological risks to plants, to environment, to human biological risk? Okay, it has to be biology related. So you cannot say about climate change. That's not biological risk. So, okay, and then for each of the risks, propose a practical approach for reducing that risk. Okay, so if you want to BS this one, the initial give you some clue, right? Like that. Okay, so if you do large scale of 1 genetic modified, this would reduce biodiversity. You can always blame on biodiversity. When you have too much of one type of plant, it's never good for the environment, right? And if you only have one type of plant, wouldn't that affect the primary consumers, the secondary consumer? Everybody up the food chain are affected. If the ecosystem is dependent on a variety of different producers at the bottom to support a diverse of animal populations. Now, because the human being comes in and decided to monoculture one type of plant, wouldn't that destroy a whole bunch of sources of food for other animals that don't depend on this GM plant? That could be a biological risk to a lot of people, right? And also we're told that these are GM modified. So wouldn't that create a biological risk that these genetic modified plants have long-term effects that is unknown on humans or animals that consume them, right? Okay, that could be one. Okay, so definitely you can just be some of them. So let's see what the answer key talk about. But remember, these kind of questions, make sure that don't contradict yourself, okay? So we talk about the risk and then a proposal to mitigate that risk. Okay, so for example, here, we have unknown human animal risk to consume them. That is a possible risk. So what is your proposed mitigation? Another point. So you can say, do more research, right? Test them, label them, so that you have a choice not to eat them, okay? You can also, there's a disruption within the food chain. Yeah. Why? Again, that because you destroy the diversity of the producers available to all the consumers, right? So how do you do that? Then you cannot, then here's a good one. It says you can intersperse GM plant, so genetic modified plant with a non-GM plant. Okay, like that. Or you can say that plant other producers so that there won't be a disrupt in the food chain, right? So that other animal that depends on other type of plant, not the genetic modified, will still have a source of food. So you can, they become very, it's not something you have to, okay? Let's see what else, yeah? You can also say that the pest, there will be a natural selection for pests that are more resistant. So there will be a resistance development in the pest species, yeah? So what do you do? To mitigate that, you can say, why don't we use more pesticides, even though that's not good for the environment. But that would be a mitigation for countering the effect of rising resistance, okay? You can introduce predators. Instead of using chemicals, you can use predators. Okay? Like if there's a rampant rat problem in Australia, you can start eating rats, right? Okay. Well, that's actually what happened in Australia, except it's not rats. They have rampant rabbit population, so they started to have rabbit meat in supermarkets everywhere to help to consume some of those rabbit population. So you can introduce a predator, or you can start commercialize them for a lot of reasons, a lot of possibility, yeah? Okay, and you can also say biodiversity. Something wrong with biodiversity because the genetically modified plant, they will outcompete other native species. So loss of biodiversity. Okay, and what do? You plant more species, right? Okay, so there's a lot of yeah. See, there's so many things you can put it in there. I don't have to go through all of them. You can say there's loss of pollinator, loss of biodiversity. You know, invasive species can wipe out the monoculture. So if the monoculture, the GM modified plant, get wiped out by one invasive species, that will collapse the ecosystem. So what do? Increase the biodiversity by introduce more non-genetically modified plant, so they won't happen. Okay, so it always have to do with biodiversity, genetic variations, and then what do you do with it? Yeah. Okay, so that's it. Super easy question. Okay, let's look at the next one. Next question is about reflex. I'm looking at here. So kind of pertinent to what we did last week. So if you look at here, the stimulus here is a nail on the finger. Yeah, that's the ancient torture. Yeah, you put the nails in the finger. Okay, or they put the nails out. Yeah. Okay, and then what is this neuron then? You can see this must be the sensory neuron, right? The receptors on your skin directly stimulate the sensory neuron to fire an action potential. Look at where it's going. It's going to the spinal cord. Now, this is a minor detail. You never have to know it for AP, but if you look at the spinal cord, all the sensory information go to the back. The back is the dorsal side. So you see the spinal cord, they will go to the back of the spine, yeah? Okay, and then you have the interneuron here, which is in the central nervous system within the spinal cord. And then you have the motor neuron that comes out in the front, the ventral side. The dorsal side are sensory input. So all the sensations, they go to the back of the spine in entering by there. And then the motor neuron comes from the ventral side, the stomach side, the heart side, yeah, ventral. So that's just minor detail. And this will go to the muscle, and the muscles cause you to have this reflex response. So it's super easy, yeah. And we said the reflex is really fast because it goes to the spine only. They don't go to the brain. They go to the brain later, so you realize it. So this is to protect you, of course. That's why we have reflex. Okay, so those people who don't feel pain, I saw was an Oprah episode that talk about people don't feel pain. It's very sad. They don't know when they got hurt. If they accidentally cut themselves, they will have to see it to know that they're bleeding. If you touch something really, really hot, you're cooking your own fingers, you don't even know unless you see it. Yeah, so it's very scary. Yeah. Okay, so definitely pain is evolutionary adaptation. So for you to stay away from something that's painful, right? All right, so now you can see this one here is also super easy. It says information processing involves complex neural pathways. Well, that's not reflex. Reflex is opposite of complex, yeah? Reflex is innate. You don't have to learn it. Everybody have reflex. Babies have reflex. They grab everything in their hands, yeah, and they suckle because that's what milk is coming, right? To signal milk is coming. They don't really have the eye reflex until later because they don't see very well until a few weeks later. So they usually respond to touch. Okay, these kind of things. Those are all reflex. Okay, they're innate responses. Reflex. So reflex is not complex. So complex one require lots of time between recognition of the stimulus and the resulting of a response, right? For some type of stimuli, a reflex replaces that typical stimulus response pathway. And the representation of reflex arcs is shown. Okay, so describe 2 ways that reflex arc differ from typical stimulus response. You see, they give you a clue, right? It says the complex neural one require time. Reflex does not. So they will always leave you a little bit clue here. You can always, if you're stuck, come back to the clue. So what are the 2-way reflex arcs different? Provide reasoning to support the claim. So you want to support the claim. What is the claim? Reflex helps organism avoid serious injury. So that's it. Yeah. Again, these are 4-point questions. So what do you have to do? So describe the difference. Why a reflex much faster? Okay. You don't integrate with the brain, does not reach the brain for response to occur. And it has much fewer synapses, right? Less distance to travel. And it's also involuntary, you can control it. So any one of those 2, yeah, 2 ways. So reflex is faster. Reflex does not go through the brain to elicit the response. That's 2 points. Or you can say reflex is involuntary and innate. It does not require brain processing. That's another point, right? It is shorter because you go to the spine, right? So any 2 points, easy 2 points, common sense. And then now you gotta support. So you gotta give a reason to support that reflex helps you to avoid injury because it's faster to a threat and response is sudden, immediate, automated. So rather than learn, right? Okay, yeah, so that's it. So it's you don't have to study the nervous system to answer this question at all, right? They didn't even ask you to identify which one is a sensory neuron, which one is a motor neuron. Nobody cares. They just want to use common sense. Okay, so again, these questions are a little bit too easy. I don't think you'll see them anymore. Okay, let's look at the next one, which is question number 7. This one here, I think is okay, and we'll do all the free response question, right? Eventually. So I just want to start this to get you guys used to these kind of questions. You always be given graph data and some kind of chart, flow chart, or some kind of biological pathway that you don't have to memorize. And your goal is to interpret what is said. So try to calm yourself down if you're freaking out, okay? So sometimes you look at the question and you just don't know what they're saying, and you have to kind of simplify. If it's something have to do with this, for example, okay, what does it have to do with, right? If you look at this question, effect of environmental temperature. So this is the effect of temperature right here on rate of oxygen consumption. So this is oxygen consumed. Okay, what does that have to do with temperature? Yeah, this is the environmental temperature, the temperature in the environment. So whatever this chart is, it's just saying that when the temperature of the environment is about 30, that's like body temperature, 38, right? 10 is very cold, 20 is still cold, yeah? So even though we use Fahrenheit, you have to know degrees Celsius. 38 is 37, 38 is the body temperature, yeah? So you can see that as the environment temperature gets warmer and warmer, what happens to your oxygen consumption gets higher and higher. Okay, we don't know why, but you can you can find a reason to say why this will be true, right? So that's what we're told. Okay, let's see what else they want. Okay, where's the question? Let me see. All right, here. So based on the graph, describe the specific method of thermal regulation used by the species of animal. So what does that mean? Yeah, that means cold-blooded or warm-blooded. Yeah. So remember the word cold-blooded means ectotherm. That's cold-blooded. Okay, and warm-blooded means endotherm. So most of the fish is cold-blooded, and all the mammals and birds are warm-blooded, right? Reptiles, amphibians are cold-blooded. Fish is cold-blooded, except like maybe tuna. Tuna is warm-blooded. So there's a lot of exceptions. And warm-blooded means that we use metabolism to keep our body temperature constant, regardless the outside temperature. The outside can be really cold, really hot. Like after today, I think the temperature will drop again. For the past few days, it was like hot, right? So our body temperature stay the same. So, and cold-blooded, the temperature, their body temperature, and so this is the body temperature. And this is the outside temperature. So this is cold-blooded. Okay? And the warm-blooded will be horizontal. Yeah, this will be warm. Okay, warm-blooded. But we're not looking at body temperature. This is about oxygen consumption. Yeah, so don't confuse these two graphs. Okay. All right. So we're looking for the specific method of thermal regulation by the animal. Provide your answer for the data. Okay. So we want to talk about, do you think this is cold-blooded or warm-blooded? If the outside temperature increases, their rate of oxygen. So what process use oxygen? Isn't it cell respiration? Cell respiration is to break down glucose for energy. So that's basically you become active. When you use more oxygen, you are becoming more active. That's the only reason you take in more oxygen. When you exercise, you use more oxygen because you need to use cellular respiration. So this is basically activity. So this is cold-blooded because when the body temperature, when the outside temperature is low, they have low metabolism, low cell respiration. When the body temperature is, when the outside temperature is warmer, then they become more active. That's what reptiles, that's what cold-blooded animals do. Okay, so this is definitely cold-blooded. So describe thermal regulation. So I would say that is not a statement. I want you to describe. I would say this organism, okay, is ectotherm or cold-blooded, right? Okay, provide support. So as the outside environmental temperature increases, the organism seems to have greater metabolic activities as more oxygen is consumed by cellular respiration. Okay, so that would be one way to say it. Does that make sense? Okay, so I'm going to say it again here. So, whoops. So again, this has to be cold-blooded because as the outside temperature increases, it seems like their metabolic rate, metabolism also increase as evidenced by oxygen consumption. The greater the oxygen consumption, the higher the cellular respiration, the greater the metabolic rate, right? So that would be your support, okay? Now, if you look at part B, now you're gonna draw a line to indicate the most likely relationship between body temperature. So now the y-axis changed. Your y-axis is now the body temperature. And then the outside temperature, this is the x-axis. So in this case, if you're cold-blooded, you got to draw a direct relationship. And so just put on slope equal to 1. If the outside temperature is 1, their body temperature is 1. It's completely dictated by the outside temperature, yeah? Because they cannot generate their own body heat very much at all. So whatever the temperature outside is their body temperature, so it will be like this, yeah? So this would be the cold-blooded. Again, it's called ectotherm, like a tortoise. I love the stupid tortoises I have at home. Yeah. Did you know that nobody knows the gender of the tortoise until they're bigger? I still don't know the 3 tortoises. I have to feed them every day. I don't know if they're male or female. Yeah. And they grow, they just, and then they just do nothing. They just come out to eat and then they go back to sleep. Yeah. If you turn on the lights, the body temperature heats up, they become active. They walk around very happy. And then if you turn it off, they just don't move anymore. Yeah. So their metabolism is completely dictated by outside temperature. Yeah. Okay. So If you are doing warm-blooded, then your body temperature will be rather constant like this. Yeah. So I'm using about 38. This will be warm-blooded. Okay. So if you ask for warm blood, it will be like this. Okay. So this is the body temperature versus this. Now, I need you to know these two in general for warm-blooded versus cold-blooded, body temperature versus metabolism. So let me go back to the notes here. Okay, so if you have this is the thermal regulation. I think it is a fair game kind of question. So once again, we're looking for either the body temperature versus the outside temperature. This is cold-blooded, ectotherm, and this is about warm-blooded, right? Endotherm. So birds, mammals are warm-blooded. Penguins are birds, warm-blooded. But if you're not doing the body temperature, if you're doing metabolism, it's weird, okay? So this is the metabolism. Also, you can talk about in terms of oxygen consumption. But that is a direct relation to metabolism. And this is the outside temperature, yeah? So we're looking at about 10, 20, maybe 30, maybe 40, right? If the outside temperature is 40, it's way too hot. So the cold-blooded one was kind of like this, yeah? Okay. Maybe like, it was like this, right? So this is the cold-blooded, okay? Now, what about for warm-blooded? Now, for warm-blooded, you got to be careful. Warm-blooded, when the temperature outside is lower than your body temperature, this is your body temperature, yeah? 37, okay? Then you actually have to spend more metabolism to keep yourself warm. You have to burn more heat, yeah? So your metabolism is actually higher and it drops, yeah? Kind of like that. And then it can go up again if it's too hot because you got to sweat. So it's not as clear, right? But definitely there's a drop, okay? So if I take that away again to make it more emphatic, there's a drop, okay? And then it should be minimized around your body temperature because that's when you don't need to burn energy to maintain body heat. But when it's really cold outside, you're losing a bunch of body heat. That's why you have to constantly eat. You have to eat more fatty food and so forth to keep yourself warm. Okay, so that's the difference there. I want to point that out. Okay. Other than that, that was it. Okay, number 7. Okay, I think that's all that they're asking. Great. Let's look at number 8. See what number 8 is. Okay, again, you only have 6 now. This is the old AP. It was too long. I think a lot of people couldn't finish, so they decided not to do this. So number 8, I'm going to transfer this one to our note because looks like here is talking about DNA stuff. Okay, genetic variation, DNA. Yeah. Okay. So every time I see the word genetic variation, these things pop to my head. Okay, I think about mutation is the ultimate source of genetic variation. If it's sexually reproducing organism, I think about crossing over, independent assortment, and random fertilization, right? Whoops, this is the wrong one. If it's asexually reproducing organism, then what do I talk about? I talk about like bacteria. I talk about transformation, right? Conjugation and transduction. So we've listed those out. Those are the sources of genetic variation for sexually reproducing organism or bacteria, asexual, right? So here is a team that have genetically engineered. So they engineered the fruit fly, okay? So what about the fruit fly? To eliminate errors in DNA replication. So errors in DNA replication is mutation, right? So is it good to eliminate error in DNA replication? Maybe, but if you eliminate errors in DNA replication, you will also eliminate variations as a result of mutation, right? So the team claims that this will eliminate genetic variation in engineered fly. So this is their big claim. Okay, so what do you think about this claim? Eliminate genetic variation in the engineered flies. Then I would say the claim is not necessarily true because there's other ways you become genetically varied, right? Like crossing over, because fruit flies are sexually reproducing organism. You can still have crossing over, independent assortment, and random fertilization. So let me just put it here. we talked about this, the sexually reproducing organism and asexually reproducing organism. What are the sources of variation? It's important we know. Yeah. So we wrote this in previous notes before. So let's just make sure you memorize this now. Yeah. You will be using a lot of these concepts for AP Bio questions. Yeah. So we talk about mutation, and mutation, there's different type of mutation. There's mutation by chance, like this one, DNA replication. There's a mutation by chemicals, and there's mutation by radiation. Yeah, all these can cause mutation to happen. They're the ultimate source. But in addition to mutation, there's other sources. You become genetically different. There's crossing over in prophase 1, so just memorize them together, of meiosis. There's independent assortment in anaphase 1 of meiosis. There's also random fertilization. Yeah. So a lot of times we don't use these 3 specific ways to describe genetic variation. We just call them genetic recombination. So you might as well call this genetic recombine, right? You recombine male and female's genes. Genetic recombination. That is a collective way to talk about 1, 2, and 3. Now, for asexually reproducing organism, we talked about transformation. Bacteria can do that. They can pick up DNA from the environment and incorporate that into their own, and therefore they can gain genetic traits, right? Genes that was not there before. Also, conjugation. 2 bacteria connect by their pili. They download each other's DNA. And also, transduction is a way to get additional genes from virus infections, right? So those are the 3 ways how bacteria do it. So if you have to talk about this, okay, first, the first claim by the researcher, okay, the team claims that this will limit genetic variation. No, right? Not necessarily. And then a second research team claimed that eliminating errors during DNA replication will not entirely eliminate genetic variation in the engineered fly. So that would be the team I am on, right? I'm on with the second team because there's other sources of genetic variation, right? Okay, so pretty easy, right? So the question wants you to provide one evidence that will indicate genetic variation has occurred, okay? So let me print this out. So what is an evidence, guys, for genetic variation? How do you know if the fruit fly is genetically different, right? So you want an evidence, okay? And that may throw some people off. So how do I know, right? What is the evidence of genetic variation? You shouldn't feel that it throws you off. So how do you know if you are a scientist, which you guys are? How do you know if there's a genetic variation? Okay. How do you know there's genetic variation in your population, right? You can look at the DNA sequences. You can look at the phenotype for morphology, morphological evidence, right? You can look at those kind of things. You can look for interbreeding, right? Are they still the same strand, right? Do they have the ability to interbreed with each other, right? So genetic variations for part A. How do you know? One evidence, that's it. Or you just have to talk about one. That new genetic variation has occurred. So you can look for morphology, new phenotype that showed up. You can sequence the fruit fly's DNA to see if the DNA sequences has changed, right? Okay. So all this could be an evidence, yeah? So let me just put it in there. Oh, good Lord. Okay. Okay. So maybe one of the days we'll have all the classes in person so I can use the whiteboard. I have not touched the whiteboard for a while. For almost 2 years. Okay. Yeah, kind of miss it, but kind of no. Yeah. It's just I have so much color here. Okay, so you can sequence the DNA. Okay. So we can sequence fly DNA to see genetic variation, to see sequences to compare sequences, okay, homology, or any change in sequences, or you can just look for new phenotypes, right? That may show. I don't like the second one because some variations don't show up as phenotype because diploidy, for example, right? The recessive allele is masked. Okay, but anyway, you just have to talk about one. DNA is the best, right? And describe one mechanism, part B, that could lead to genetic variations. Okay, so we talked about other than DNA replication, it could be crossing over, right? Independent assortment, just one. But if you just say crossing over, do you think you get 1 point for part B? When the question asks you to describe the mechanism, not to state, right? So no, so you want to talk about something, right? So how do you make this a description other than a statement? You can say that crossing over in prophase 1, right? Between homologous chromosome, yikes, between homologous chromosomes, shuffles DNA, and generates variations, right? So you can say that. So you can also talk about independent assortment, then talk about that the homologous pairs separate independently of each other pair, right? Of other pairs. So this will generate different gametes as different alleles are being separated into different gametes, right? You can also talk about random fertilization. So part C, how genetic variations in a population contribute to the process of evolution. So this is Darwin. Well, genetic variation is super important for Darwin, right? In fact, if there is no genetic variation, there can be no natural selection. So how genetic variation contribute to the process of evolution? Yeah. So you can say that genetic variation is the foundation for natural selection to occur, right? In order to evolve, there has to be a differential reproduction. So some members have to adapt better to survive to pass on the gene, and that is not possible unless there's genetic variation, right? Okay, let me just type that out. That might be too long, but you know, when you answer these kind of questions, you want to shorten your response as much as possible. So I can say that the variation, okay. What did I say? I forgot. So variations are foundation for evolution by natural selection. How about that? For evolution by natural selection. Okay. And then okay. I'm gonna say that variation, or I can skip that sentence. I just said variations allows for differential reproduction of more okay. Variation allows for differential reproduction of the fittest members, right? Fittest individuals. Okay. And without it, without variation, no natural selection is possible. Okay. Anyway, something like this. Let's look at the scoring guide, okay, and see what they'll take points for. It's, you know, you don't have to be 100% happy with your response, okay, but let's see what the scoring guy would say here. Okay, so again, this is question number 8. There's no more question number 6 and 8. The old questions are 3 points for question number 8. Yeah. So here it says one evidence. So you can talk about, are there new phenotype? Yeah. And that's it. Provide one evidence. So you don't have to really explain too much. Is there new phenotypes in the fruit fly, right? There are new genotype. What the heck? Different DNA sequences or sequence of DNA. Is there a chromosome difference? It's the same thing, right? Okay. You can even look for different mRNA. Different proteins are produced by the fruit fly. Yeah, they're really stretched, but they will just want to give you the point. They want to give you the point. You just have to say something. All right. Next one. One mechanism that lead to genetic variation. So all this, look how much they can talk about. Sexual reproduction, recombination to produce new allele, new traits, meiosis, produce recombination, crossing over, produce new recombinations, independent assortment, and random fertilization produce new combination alleles and traits. So the reason that new combination is because the shuffling between maternal, paternal, homologous chromosome. Yeah. So any one of those. You cannot talk about mutation. Yeah. Because this is other than mutation, right? Okay. All right. And then part C. Wow, there's more. Holy moly. Okay. So there's more you can talk about. Gene flow, oh, that is stretch. I would not talk about gene flow. It doesn't talk about there's a chance of gene flow, new allele. But that could lead to variation if the gene flow into this population. That is very stretched. You can talk about non-disjunction, which is meiosis error, right? You can talk about chromosomal mutation, the large ones, like you can talk about inversion, duplication, right? Translocation of a piece of chromosome to another non-homologous chromosome. Those are huge ones, right? You can also talk about chemicals cause mutations. Yeah, you can talk about mutation. I guess you can, yeah, even though it's not backed up. But that's not the one that I was thinking of, yeah? Okay, part B, part C. So the last one is how genetic variation contributes to natural selection evolution. So genetic variation is the basis of phenotypic variation that can be acted upon by natural selection. And without genetic variation, there's no variation on which natural selection can act on. Yeah, so basically that's what I was trying to say. This is probably much better worded. It's just in one sentence, yeah? Without genetic variation, everybody will be identical, so there's no selection for more fit individuals. Therefore, there's no natural selection, and therefore there's no process of evolution. So one thought leads to the other, yeah? But try not to repeat this, yeah? Okay, you cannot just write an answer that repeat this. For example, some people will say that evolution requires genetic variation. You get 0 points by saying that. That does not answer any question. Evolution requires genetic variation. No, there's no answer there. That's a BS. That is not very well done. So you want to describe how, right? How genetic variation. Genetic variation provides the basis for natural selection to act on, so that there's always members that are different enough to survive, to reproduce, to pass on the traits for evolution to occur. That would be okay. Alrighty. So these are the points. So what I did not ask you guys to do is to actually add up all your points. I know some of you guys turn it in, like 4 or 5 people turn it in. So in the future test, when we do full response, I actually want you to grade on the side so you can add up all your points, and we're going to put it on the Google Classroom, yeah? So that we know that what point you got. So since this is the first test, I just want to introduce you some of the rubrics. This is kind of scoring guide you're looking for. They're usually not too bad, but some of the scoring guide is not very good, yeah? So again, next week is also free response. We're going to do several free response, get used to, and then we have the multiple choice coming in, yeah? So the key is not to memorize a whole bunch of, biological facts, that's not how the test is. So you have to really remember that. A lot of them is common sense. If you look at some of the questions that we just did today, if you were a English major in college, or if you love science, but you're not necessarily taking AP Bio, do you have to read ecology to answer this question? No. Do you have to know the entire nervous system about what neurotransmitter does what to answer the reflex question? No. Do you have to know classification taxonomy to answer the cladogram, to build a cladogram? The cladogram is the easiest question you'll get, okay? But it has to do with evolution a little bit, okay? And same thing here. Do you have to know, then this question has to do with common sense, right? The selection in the presence of predator. That is probably one of the most common one. Predator, no predator. And what does that do to a population? Yeah. And then this is a GM modified plant. I don't think we'll see those questions anymore. Reflex is way too easy to talk about. But I think it is important about the immune response. Okay. So if you memorize that, immune response, different stages of specific immunity, you can talk about any one of those bulleted points, yeah, as potential stages in the activation of specific immunity. And the last one has to do with genetic variation, yeah, without going into detail about meiosis, mitosis, okay? So these kind of questions show up later on in 2016 again. They want you to draw meiosis, mitosis out, and then talk about the kind of gametes that would form, right? Dihybrid cross, stuff like that. So they can combine meiosis with genetics, with meiosis with genetics. Okay, all right. So this is overall a pretty easy AP. Definitely not hard. I always said the hardest question is the first one, because you're giving something about trichome and what the heck, you got to talk about dependent, independent. But every time you talk about an experiment, know about, well, what would be the null hypothesis for this experiment? Because this is not a null hypothesis right here. We're given one hypothesis, which is not the null hypothesis, right? Well, our hypothesis is that higher trichome density are selected for. That's hypothesis. So if this were to, if the question were to ask you for the null hypothesis, then you should say that there's no difference, right, in the trichome density. Sorry, I should say that in the presence of herbivore, so you have to think about that there's no difference in trichome density, whether there's no selection for higher or lower trichome density in the presence of herbivore, right? That would be the null hypothesis. The null hypothesis is always that there's no preference for either one, for low or for high, right? For head or tail, right? So everything is the same, yeah? So if your chi-square test indicates otherwise, then you either accept your null hypothesis or reject your null hypothesis. If your chi-square shows otherwise, then you got to reject that null hypothesis. Indeed, there is a difference that higher trichome density are indeed selected for when there's herbivore. So you reject your null hypothesis. So you support this hypothesis. Okay. So if you're given no hypothesis, then most likely you'll be using data to reject it. If you're given hypothesis, then you have to find data that supports, in this case, yeah, the hypothesis. So is that okay? So don't be careful about hypothesis versus no hypothesis. Okay. So we'll do more of those, of course, and then we'll stop here then, guys. Thanks everybody for coming in. I'll see you guys next time. Yeah.